The topic of this lecture is capillary fluid dynamics. And what that means is we'll discuss what causes fluid and solutes to move between the capillaries and systemic tissues. The direction, the quantity, and the forces involved. So first we should recall the flow of blood in the capillaries. We know that in the capillary beds, velocity of blood flow is very slow. This is partly due to the fact that there's small vessels, so more resistance, but also because there's a large cross-sectional area through which blood can flow in all the individual capillaries in the entire capillary bed. This slow velocity is an advantage for the exchange of nutrients and wastes between the blood and the systemic tissues. So materials are exchanged across capillary walls from the blood or plasma out to the interstitial fluid, as well as from the interstitial fluid back into the blood. Some things that are exchanged include gases, solutes, and water. What's typically exported out of the capillaries would be oxygen, nutrients like glucose, amino acids, lipids, calcium and minerals are important, and antibodies can actually leave the blood as well as hormones to get to their target cells, and electrolytes and ions kind of goes with minerals. Albumin typically is not exported from the capillaries, but it is present in the capillaries, and I think it's really important to note that it's such a large protein that typically it can't get out. That can only happen in a diseased state. But also what is exported from the blood is water. Some things that are imported into the capillaries would be carbon dioxide, because the tissues have created a lot of carbon dioxide in the process of making ATP, and that needs to be picked up by the blood. And then glucose, depending on the concentration of glucose in the tissues, it might be needed elsewhere. Like when the liver makes extra glucose from gluconeogenesis, that needs to be transported to other locations in the body. Certain fatty acids can leave one tissue and go to another, and they need the blood to do that. Again, calcium and minerals, antibodies, and hormones as well. And what we're really going to look at is fluid movement. So we'll see here that water not only can leave the blood and go to the tissues, but it can leave the tissues and go into the blood. Okay, let's review the various types of capillaries that we already discussed. There are continuous capillaries, fenestrated capillaries, and sinusoid capillaries in the body. Each of these are in different locations. The most common are continuous capillaries, and that's what we're going to deal with here today. They are the least permeable due to the fact that there are tight junctions between the endothelial cells, and there are parasites, these cells on the outside, with foot processes that prevent the escape of materials. Fenestrated capillaries are more porous, so there would be more leakage from the blood out. There would also be, of course, more import, but due to blood pressure, the danger would be more leaking. But it's an advantage in certain locations, like in the kidney that needs to filter blood. We'll see that there's fenestrated capillaries in that location. Sinusoid capillaries are very super leaky, And these are located in the bone marrow primarily. At least that's what we'll discuss. And again, in this lecture, we're going to deal with just the basic continuous capillaries, which are the most common throughout the body and the least permeable. So if we look at the wall of a continuous capillary, we see that there are various locations through which solutes can move. Between the cells, intercellular junctions, sometimes they're called intercellular clefts. Across the cell membrane, if there's a channel for the substance, or maybe the solute is lipophilic and it can cross the bilayer, the phospholipid bilayer. And then third, in fenestrated capillaries, which we won't really see in continuous capillaries, substances can move through those wide slits called fenestrations. And the last way that solutes move in and out of the blood are through vesicles, or inside vesicles, through exocytosis and endocytosis. So primarily what we're going to deal with is fluid movement that occurs between cells as well as across the membrane due to the presence of aquaporins. Now, just because there's a location for something to move, that doesn't necessarily mean that it will. Instead, the same transport mechanisms that we studied in A&P1 are involved. So any substance that's going to move could be through various processes. One, it could be diffusion. Two, maybe there's active transport, particularly when going across the cell membrane. And filtration is a concept that is a little bit like diffusion, but there's no concentration gradient per se. So what happens instead is the force, physical force, mechanical force of a substance will force it out of the capillary wall. And then finally, another transport mechanism by which solutes move is endocytosis and exocytosis through these vesicles. And I already mentioned that. So there is a distinction between the route something takes, meaning the location, and the mechanism that governs why it moves. In some cases, it will move due to diffusion, in some cases, active transport, and sometimes by a physical force called filtration. We are going to deal with mainly filtration and diffusion. Okay, let's look at water movement. Okay, so those were solutes. Now, water can move primarily between cells and across the cell membrane, aquaporins, and through fenestrations if they exist. And the process by which most water moves out of the capillary bed we will see is filtration due to the force of the blood pressure. And so filtration actually can happen through cells and through fenestrations. I mean between cells and through fenestrations, either one. But water will also move by osmosis, and that will depend on a solute concentration gradient. And remember, osmosis is water diffusion from an area of low solute concentration to an area of high solute concentration. And so osmosis could happen at any of these locations. You may have read in your textbook about a concept called bulk flow, and this refers to fluid movement. And It turns out that approximately 20 liters of fluid moves out of our arterial capillaries, arterial end of the capillary bed every single day. That's a lot. That's like 4 times your blood volume. But most of it will return to the venous end of a capillary bed. And so these are the dynamics we're going to assess. Why does so much fluid leave the capillary bed only to come back into it? Okay. But it is important because this movement determines the amount of fluid that's in the bloodstream as well as in the interstitial space, because we need our body tissues to be bathed in fluid as well. Okay, now we're going to look at just fluid movement. And these are the two forces that contribute to the movement of fluid, or we'll just call it water, in or out of a capillary. One force is osmotic pressure. Water diffuses to a region of highest solute concentration. And so there is a gradient of solute concentration between the plasma and the interstitial fluid. And whichever is higher is the direction that water will move. So this force is referred to as osmotic pressure. Another force that contributes to water movement is hydrostatic pressure. And this is due to the accumulation of fluid in either location, inside the capillary or out in the interstitial space, that creates a force on the vessel wall. So this hydrostatic pressure will differ in the interstitial space and in the bloodstream. So wherever it's higher, that will force the water to leave or to move in or out. We refer to this force as hydrostatic pressure. Now, hydrostatic pressure in the plasma is just what we've already learned about. It's blood pressure. Okay, now before we get into details about what causes fluid to leave the capillaries, either osmotic pressure or hydrostatic pressure, and then re-enter the capillaries, osmotic or hydrostatic pressure, there's a couple of terms that are commonly used, and we need to learn what they are. So let's keep in mind that there's always a balance between osmotic pressures and hydrostatic pressures in the interstitial space and the capillary, and those determine the amount and the direction of fluid movement. So there's a term called filtration, and filtration is really reserved for fluid moving out of the capillary due to whichever force, osmotic or hydrostatic. But typically, it's a hydrostatic blood pressure cause. So here's filtration. Water is leaving the bloodstream. And then another term that we'll encounter is reabsorption. In this case, fluid moves into the capillary from the interstitial space, and this occurs at the venous end, which we've already kind of talked about a little bit. So what we see overall is at the arterial end of a capillary bed, we see filtration, a net filtration versus reabsorption. But at the venous end, we see a net reabsorption. And 85% of the water that is filtrated out of the capillaries will be reabsorbed by osmosis back into the venous end of the capillary. And later we're going to talk about, well, what happens to the other 15% that's not reabsorbed. But for now, let's talk about how that 85% gets out and back in. Okay, just focusing on the arterial end of the capillaries, let's look at what forces are involved to cause net filtration. There's hydrostatic pressure of both the blood, labeled A, and interstitial fluid, labeled B. So a hydrostatic pressure is against the wall of the vessel, and we will use blood pressure, this is a value in your textbook, to be the hydrostatic pressure in the capillaries. So 35 millimeters of mercury is an outward force for fluid to leave the capillaries. But there is a fluid accumulation in the interstitial space, and that's pretty negligible. Even if it's not zero, it's very small, but it would be an inward force if it existed, but it really doesn't play a big role. Now, the osmotic pressures at the arterial end do play a role in both the blood and the interstitial fluid. So osmotic pressure is mainly due to solute concentrations, and in the blood, this is mainly due to albumin, because albumin is such a large protein that doesn't get filtered out. So albumin attracts water. So water from the interstitial space will come back into the blood because water will diffuse to that higher concentration. Now, the interstitial space has an accumulation of some solutes. Just think about CO2, for example. That's a very small molecule, but there are other things there too. Lots of proteins and cells and everything. So that has a very small force, osmotic force, but it would draw some water out of the capillary bed. So what we see is that there's two forces that play a role in water movement: hydrostatic forces, forces of fluid, and osmotic forces, which is due to solutes. And at the arterial end, the biggest players are blood pressure, which is a hydrostatic pressure. That's an outward pressure. And osmotic pressure of the blood, because there's these large proteins in there, and that's an inward pressure. So let's just kind of assess what are the values and which direction do we expect water to move, the bulk of water to move. Where's it going to go? Well, if we just look at what are the forces that cause water to leave the blood, outward forces, we see that there's two. The hydrostatic pressure of the blood, which is A, and this very small osmotic pressure of the interstitial fluid. Those are the two outward forces. And so the total of those are 36 millimeters of mercury. But there are also inward forces, of course, and the inward forces include the hydrostatic pressure of the interstitial fluid, which is basically negligible, none, and then the osmotic pressure of the blood due to that presence of albumin. And that's a large consideration. And so in total, the inward pressure for blood to go, or water to go back into the blood is 26 millimeters of mercury. And so outward minus inward leaves us with a net outward movement of fluid at a value of like 10, pressure of 10. So the net filtration pressure at the arterial end of a capillary bed is 10 millimeters of mercury. And the biggest reason that there's net filtration is that high number of the blood pressure, the 35 millimeters of mercury for blood pressure at the arterial end of a capillary bed. so water goes out. Okay, now we're going to look at the venous end of a capillary bed. And what are the forces that are happening there, and what are the values of the forces? So for hydrostatic pressures, we have a hydrostatic pressure of the blood, which is a little lower at the venous end, like half. So that has to do with the fact that, remember, blood moves slowly through the capillary bed, right? There's less force. And so that's 17 millimeters of mercury outward force, labeled A in the diagram. And the hydrostatic pressure of the interstitial fluid at the venous end evidently doesn't change a lot. They say it doesn't change a lot. I have a hard time believing that, but that's okay. If we just had a lot of fluid leave the capillary to bathe the tissues, I would think there would be more fluid here, and this inward force would be a little bit higher, but they say that it just stays the same and it's still 0. So we don't consider it. Fine. It won't make a difference for what we end up doing. For osmotic pressure, we still have that albumin in the blood. It never left. So there's still 26 millimeter of mercury pressure due to the presence of albumin in the blood that would cause water to move by osmosis back into the bloodstream. But there are wastes in the interstitial fluid that cause water to leave as well, but it's a very low number, and it's D there. So just like we did at the arterial end, we're going to summarize all these forces by figuring out which ones are outward forces and which ones are inward forces and what the values are. So we're at the venous end of the capillary bed, and there's 2 outward forces for water to move. One is still hydrostatic pressure of the blood, or blood pressure. It's lower though, it's not 35 now. Now it's 17, and that's A. And then there's osmotic pressure of the interstitial fluid, which is D here. And that's minimal, if you remember. So both A and D contribute to an outward force of water movement. Now there's also a couple of inward forces causing water to go into the capillary, and that's hydrostatic pressure of the interstitial fluid, which supposedly is 0, and osmotic pressure of the blood due to that albumin of 26 there. Okay, and so if we add these together, we see that there's an outward force for water movement of about 18 and an inward force of 26. So the inward forces are greater here. And so that's why water moves back into the venous end of a capillary bed. In total, we see that net reabsorption at the venous end is 8 millimeters of mercury. So fluid will go in. And this is primarily due to the osmotic pressure of the blood. The proteins that remain in the blood and have never been filtered out at the arterial end, they're still in there. And so they draw the water back in. So in summary, we see fluid exchanged between the blood and the interstitial space by filtration at the arterial end, primarily due to high blood pressure, higher than at the venous end anyway, and water moves back in due to the process of reabsorption, which is really osmosis, and that's due to the presence of proteins, plasma proteins in the blood. One thing to note, that at the venous end, blood pressure is low, right? It was 17. And it cannot, I can't draw that, I'm trying to. It cannot counteract the osmotic pressure of the blood. So remember, blood pressure is an outward movement. So 17 out. Man, it's annoying. And the osmotic pressure inward is 26. At the arterial end, of course, the blood pressure is so high that the osmotic pressure of the blood is overcome, and 20 liters per day are filtrated out of the arterial end of the capillary. And at the beginning of the lecture, I said that 85% of the 20 liters is going to be reabsorbed at the venous end. That's 17 liters. I did the math. So what happens to those 3 liters that aren't reabsorbed? Does that fluid just stay in the interstitial space? Well, some of it does, right? But what that allows for is refreshing the interstitial fluid. And the 3 liters, maybe the same 3 liters or at least 3 liters, are going to be recaptured by lymphatic vessels, and they're green in this picture. So any water that's filtered out of the arterial end will bathe the tissues, and the same amount will be captured back into the lymphatic system. At least the fluid that's not reabsorbed. So let's look at what happens to the fluid that's traveling in the lymphatic system. The lymphatic system has a connection to the circulatory system. So lymphatic vessels lead to ultimately a connection with 2 veins. And these veins in the circulatory system are called subclavian veins. And so the lymphatic vessels have ducts that allow the fluid to go back into our venous circulatory system. The 2 ducts are called the right lymphatic duct and the thoracic duct. And we will do the lymphatic system again later, and those names will come back and we'll cover them again. For now, I'd like you to know that the fluid recovered by the lymphatic system is returned to our venous blood circulation. So it gets back into the blood, just not right away at the capillary bed. Now we're going to look at some disorders associated with these fluid dynamics at the capillaries. One is hypertension and the other is hypoproteinemia. So I'm going to do hypertension first. Recall that the definition of hypertension is chronic high blood pressure at a value of greater than 140/90. And chronic meaning all the time. Now, the problem with this high blood pressure or hypertension is that it makes the heart work harder. There's increased resistance. The vessels are probably narrower. The blood is pushing against the wall of the vessel harder, but it encounters more friction in doing so, so resistance increases. And we also call this an increased afterload. So the heart has to work harder to counteract this afterload or this resistance. Now, there really are 2 causes or types of hypertension. One cause we just categorize as primary hypertension, and it's due to the condition of the vessels themselves. Secondary hypertension results from a different disorder, like lung dysfunction that causes changes in the viscosity of blood or blood volume. And kidney disease is another example where the blood can't be filtered, and so there's an accumulation of solutes and fluid in the blood. So we'll deal with primary hypertension first. Primary hypertension is due to the wall of the arteries primarily being narrowed or thickened, and that increases afterload, so the heart has to work hard. Now, the risk factors for developing primary hypertension are obesity, sedentary behavior, tension, what you eat, which relates to obesity, and nicotine can cause hypertension because it causes vasoconstriction. Now, the results of this primary hypertension, this high blood pressure, will be edema because the net filtration pressure will be higher. Another factor, or another effect, of hypertension is that the heart is working harder and it hypertrophies, meaning the wall of the ventricles gets thick. Over time, that thickness actually becomes stiffness, and therefore the heart is inefficient over time and can lead to heart failure. Now, what's wrong with the vessels? I know I mentioned that there's narrowing of the vessels or some sort of blockage. Well, there could be a tear or some kind of a lesion in a blood vessel that could be caused by hypertension, but even if it was caused by something else, it becomes a site for repair and the development of atherosclerosis. And so atherosclerosis creates hypertension, the narrowing of the vessel. Where there are these formations, the sclerotic or hardening regions, you often get kind of a breakage of the lesion, and that weakens the wall and can cause an aneurysm. And we talked about aneurysms when we did the blood vessel lecture. Now, sometimes when the vessel wall becomes thickened, the things that are supposed to come out of the blood don't, particularly in the kidney. So it might result in kidney disease. And certainly, if the vessel blockage is severe enough, you could have ischemia to tissue, which means a lack of blood flow, and consequently low oxygen or hypoxia. So what you see in terms of edema with chronic hypertension is an increase in the amount of interstitial fluid. It's due to this increase in the outward hydrostatic pressure of the blood in hypertension. And so you get swelling in the tissues. And I have a picture here of severe swelling. We call it pitting edema because if you press on the skin and then you remove your thumb or finger, whatever, a pit will remain for a certain period of time versus go away rather quickly. Now we're going to see that this kind of edema can also happen not in hypertension, but it can happen in malnutrition. And that cause of edema is a decreased osmotic pressure of the blood. And we'll come back to that a little bit later. So how is hypertension treated? I guess it depends primarily on the severity, right? But one of the first things that usually is done is the application of diuretics. When a patient takes diuretics, they urinate more. Okay, diuretics act on the kidney. And so if they urinate more, they lose water or blood volume, and that decreases the blood pressure on the vessels. There are some other things that can be tried. Calcium channel blockers, but that is mainly to protect the heart muscle so that its contraction isn't so severe that it develops hypertrophy. And then there are also these drugs called ACE inhibitors and beta blockers. And what they do is block the effects of angiotensin II. And remember, angiotensin II is a vasoconstrictor. Now, sometimes if chronic hypertension is caught early enough, just behavioral treatment will work, like weight loss through exercise, changes in diet, and stress reduction. Now that's for primary hypertension. What about secondary hypertension? Like in the case of lung disease, where the reason the blood is more viscous is because there's lung disease, and that causes the hypertension. Well, you have to treat the lung disease. Or what if there's primary kidney disease? There's several genetic kidney diseases, and if the kidney's not filtering the blood properly, then that's what has to be treated in order to alleviate the hypertension. Okay, let's look at what we just learned about regarding the forces of capillary fluid exchange in the capillary beds and see how hypertension results in systemic edema. Okay, what are the forces that change and why does edema occur? Well, I'm going to give you the answer here. This is normal numbers. But if we look at what happens in hypertension is that the blood pressure goes up both at the arterial end and at the venous end. So that would be a higher outward pressure at both locations. And so you'll have less reabsorption as well as higher filtration. So increased filtration occurs and less reabsorption occurs. That causes edema. Okay, that's our first disorder. Now let's look at the second disorder, malnutrition, specifically hypoproteinemia, which refers to low protein in the blood. Now, why would there be low protein in the blood? It can be due to liver disease, because remember, that's the site where many plasma proteins are synthesized, including clotting factors, albumin, everything. There's a lot of things made in the liver. Or it could be due to kidney disease, where the kidneys are filtering out the protein. The protein's going into the urine. But in our case, we're going to look at malnutrition, starvation, and in that case, you have lower amino acid intake. Again, you're going to see edema, but you'll also see muscular atrophy because there just isn't enough protein to keep the muscles in tone. All right, let's analyze what we learned about the forces in the capillary bed causing fluid movement and which forces change in hypoproteinemia and why does edema occur. What changes is that there's a lower osmotic pressure in the blood at both the arterial and the venous ends. So we before had a blood osmotic pressure of 26, and now that's going to be lower in both the arterial end as well as in the venous end. So what happens is if you have less osmotic pressure at the arterial end, less of a force, that will increase the filtration at the arterial end. And if you have less osmotic force at the venous end, then you'll have decreased inward reabsorption. And so that will result in edema because less fluid will be reabsorbed. Now there's many other causes of edema, but these are the ones that are I think the most relevant to capillary fluid exchange. Other causes of edema could be the blockage of lymphatic vessels, because remember, it's the lymphatic vessels that capture that 15% that's not reabsorbed at the venous end of the capillary. And liver disease we've already talked about, so that one's not new. Kidney disease, we've talked about that one. But another one is inflammation, which is, it might be a normal situation, right? Inflammation due to injury, infection, whatever. Inflammation increases the permeability of capillaries so that the tight junctions aren't quite so tight. And therefore you get more fluid escaping the capillary at the arterial end, and you might even get some proteins leaking out of the capillaries. And so you get swelling. But hopefully this is temporary, right? Because eventually the water will be captured, and the proteins will be transported back into the blood or broken down into amino acids. Another one that relates to what we talked about when we did histology of the vessels is the incompetence of venous valves. We talked about varicose veins in particular. And when blood pools in the capillaries and stays there for a while, and the veins as well as the capillaries kind of distend, then more fluid will leak out. It's a little bit like having leakier vessels, but it's due to a hydrostatic pressure. And congestive heart failure is another situation that we've seen where edema develops. And it has to do with the flow of blood backing up either in the systemic circuit or in the pulmonary circuit. So that is the end of our lecture on capillary fluid dynamics. Thank you very much for listening.